All right, so in part three, what we're going to talk about is a little bit how the cell cycle is controlled. So when in the 1970s, when the scientists were learning a lot about like what controlled cell division, and they really started to kind of look at cancer and what it was and what caused it and things like that, they did a bunch of different studies and experiments and things like that to try to determine what it was that was causing the runaway growth in cancer cells, what it was that was causing just cells to divide normally, and a whole bunch of other things. And they came up with what they termed the cell cycle control system. And so what this system was really is a way in which the cells kind of go through a repeatable cycle of events that's going to control the cells. And what they found when they did some of these experiments were there were some things that existed in the cell during a particular point in time that were controlling the events of the cell. So for instance, in this first experiment that you see here, when they had a cell that was in the S phase, it was synthesizing its DNA. No matter what type of a cell they fused it with, they got the cell to enter the S phase and start replicating its DNA. Okay, so these cell fusion experiments, like in experiment 1 here, they fused an S cell with a G1 cell, and they both began or continued synthesizing the DNA. If they had a cell that was going through mitosis, didn't matter what the other cell was that they fused it with, that cell would begin dividing itself as well. So in other words, this G1 cell, it hasn't replicated its DNA, it shouldn't divide, but when you fuse it with an M cell, you can get it to divide. And of course, that's going to be an abnormal cell. So that was a little bit puzzling. And what they tried to figure out was what it was. Why was this happening? What was causing this thing? And what they found is that each of these cells had these substances that were in them that would control the movement from one stage to the next to the next. And what you can also see too is that these cells are controlled by a series of checkpoints within that cell cycle. And there's a lot of cytoplasmic signals that are going to control those signaling pathways. And what they termed these things were cyclins and kinases. These are the two main types of regulatory proteins that you find inside the cell. And they work together to basically kickstart certain events in the cell, such as DNA synthesis, cell growth, all of those types of things. And cyclins, so you can think of like a cycle, something that repeats, something that rises and falls, things like that. Cyclins are the substances that are going to rise and fall throughout the various stages of the cell cycle. Okay. And they're going to interact with kinases, and the kinases are present in basically a constant amount in the cell throughout the life of the cell. And as the kinase levels rise and they interact with these cyclins, they're going to cause a whole variety of these changes. So if you look at this graph right here, you can see that the various cyclins levels kind of increasing and decreasing through the different phases of the cell cycle. Okay. So you can think of cyclins again. These are a family of proteins that are going to interact with kinases. And once they do, they're going to activate a series of enzymes that's going to turn on this signaling cascade that's going to result in the cellular response, whether it's DNA synthesis, protein synthesis, or whatever. That's how these things are going to work. And again, the kinases, these things are going to be present again in a constant amount. And what they're going to do is they're going to function by either activating or inactivating other types of proteins through phosphorylation. Okay, so they're going to add a phosphate group on, typically activating the protein, but there are times when they can inactivate them. And the activated proteins now, these are the things that are going to serve as those go signals and kind of get that cell to go through the different phases of G1, S, G2, and M, and so forth. Okay. And as I mentioned a couple of times already, they're present, the kinases are in constant amounts. And when the cyclin level rises during that particular phase of the cell cycle, it interacts with the kinase. That's when it's going to kind of get the protein kind of doing what it needs to do. These things are often dubbed CDKs for short, cyclin-dependent kinase, and these are the things that are going to trigger the events of the cell cycle. Okay, so usually what happens when these things interact is you get a shape change in a protein, and that shape change is what's going to lead to a different, outcome or different event in that particular cell cycle. Like we said, increasing the amount of phosphorylation that is going to activate these proteins. For instance, in this example, if we're activating the proteins that are involved in helping those chromosomes condense down, that's an important step in making sure that we're going to evenly distribute all of the genetic material throughout the cell during cell division. Other types of these CDK are going to be involved in breaking down the nuclear envelope, like in prophase, and so on and so forth. So there's a number of different things that these things can be involved in here. Okay. And so if you look here at the example that we've got, and this is just one example of many. You can see the green, the M cyclin right here, as this thing is going to interact with the kinase that's already there. It's going to promote the events of the M phase of the cell cycle and mitosis. But once we reach the end of mitosis, now these cyclins start to get broken down. And then we have another kinase that's going to be present in the cell. Again, there's many of these things, and this is going to interact with another particular type of cyclin. In this case, the S cyclin, which is going to be involved in like synthesizing the DNA. And so once this thing is done synthesizing the DNA, it gets broken down, and then there's another interaction that's going to just take it through the varying events. Okay, so for simplicity's sake, what we're just showing you here is just a couple of examples of which, you know, there I'm sure are hundreds of them in the cell that are taking place that are involved in regulating the various stages of the cell cycle. So there's 4 major classes of cyclins. I don't know exactly how many total cyclins there are, but there's 4 major classes, and you have G1s cyclins, and these are going to bind to the cyclin-dependent kinases here at the end of G1. These are going to commit the cell to replicating its DNA. Okay, then you have some S cyclins. These are the ones that are going to rise during the S phase. These are necessary for the various aspects of the DNA replication. You have M cyclins that are going to promote the events of mitosis and all of the things that we've kind of talked about in an earlier lesson. And then the G1 cyclins are going to, you know, push the cell through the G1 phase of interphase. So certain cyclin signals are going to shut down certain aspects of that cell cycle, okay, and allow other ones to kind of get going and start up the other parts of it. So there's a bunch of, or a few, I should say, checkpoints within the cell cycle. You have the G1s checkpoint, the G2M checkpoint, and then there's an intra-S phase and an intra-M phase checkpoint. And these are just checkpoints to make sure that everything is being handled correctly in terms of the DNA, so that we're not copying faulty DNA. Because if you recall from an earlier discussion, we said that we want to produce these exact copies, and the cell has ways to ensure that we're producing exact copies, and that's what these various checkpoints do. Okay, so the first checkpoint, the G1s checkpoint, is usually known as like the restriction point, because at this point, if you don't get a go signal, the cell is going to either enter G0, or it's going, the body's going to get rid of it through apoptosis or something. And if it does receive that go signal, then it's going to synthesize its DNA, pass through G2, and divide the cell if everything goes right. Okay. And what the cell's doing here really is it's checking for the appropriate size to make sure that it should be dividing, looking at the nutrients, making sure those are available so that it can have what it needs in order to divide, and checking the DNA integrity and all of these types of things. And again, if it doesn't, it enters G0. G0 is basically called quiescence. Sometimes it's a state of non-division, and muscle cells and nerve cells are things that are common to G0. Okay, liver cells, that would be another common one. Those are 3 common cells that are in G0. They're not dividing very often. But usually during G1 now, you've got a lot of these cyclin kinase interactions that are going to promote DNA synthesis in the S phase. And if there's any DNA damage that occurs, like from sunlight or from chemical exposure or something like that, you're going to slow down the cell division. You're going to try to fix these problems. Okay, and that's going to halt that cell in G1 while these things get fixed. And if they can't fix them, like I said, the body's going to get rid of it. Some of the repair mechanisms that we'll talk about in a later discussion involve P53, and that's one of the kind of the guardians of the genome, one of the things that's going to be involved in really making sure that we can repair that DNA, kickstarting a bunch of different mechanisms within the cell to ensure that we can fix the problem, and then ultimately, if not, kind of kickstart the pathway that leads to apoptosis and the destruction of the cell. So I mentioned a minute ago this idea of G0 or quiescence. So what's happening here is these cells are not in a state of division. Okay, so they're actively going through interphase. They're doing the things that they need to do. And the only main thing they're not doing is dividing. So mature mature muscle and nerve cells don't divide. They're just doing their job. Some cells are in G0, but they can be stimulated to divide. So in other words, like an example that's commonly given involves your liver. If you get some liver damage or you get an accident or something like that, oftentimes the liver can kind of regenerate itself in a way that can repair the injury and stuff. All right. So now the intra-S phase checkpoint, this is a checkpoint that's kind of the surveillance camera during DNA synthesis. So it's going to make sure that the DNA is being replicated properly, and it has the ability to kind of stop the process and fix the DNA if there is an issue. You know, it'll prevent it from going into G2. And again, if you can't fix it, it'll kickstart apoptosis. And basically what's happening is you'll learn about a replication fork when we talk about DNA replication. And when this thing encounters damaged DNA or some sort of an error, that's going to get a series of chemical reactions going that's going to stop mitosis. It's going to stop DNA replication, and it's going to attempt to fix that problem. Okay. And again, as I mentioned a couple of times now, apoptosis will ensue. That's programmed cell death if you can't fix the damage to the cell. Okay, so now during the G2M checkpoint, this is the damage checkpoint. Again, the cell is going to check for damage because we don't want mutant cells. We don't want damaged DNA being replicated, those sorts of things. And if any errors happen to get detected, you get a variety of CDK pathways that are going to hold that cell into G2 while you can fix the damage. During G2, you get a lot of these M-cyclin molecules that are going to start to interact and promote entry into the M phase during mitosis. And the interaction of these particular cyclins at G2 are going to form what's called maturation promoting factor, or MPF. And this stimulates the entry into the M phase. And high levels of cyclins now associate with these CDKs, and this is going to give you this active MPF complex. This is the thing that's going to promote all of the events that are leading to mitosis, the breakdown of the nuclear envelope so that the mitotic spindle can form, the condensation of the chromosomes that you see during prophase. All of these different things are going to be kind of governed by this MPF, okay? And so you can see here the levels of MPF activity are going to rise during the M phase and then fall off real fast. So those are the kind of the chemicals that are associated with mitosis. And then one of the unique things about this is that the MPF has a hand in kind of controlling itself in terms of being able to switch itself off. And what it does is it basically kickstarts a process that leads to the destruction of the cyclin that activates it. And then what happens is you know, that kinase, that CDK, that kinase is going to remain in the cell, and then it'll become a part of that MPF complex again during the next round of the cell cycle. So it has a way to kind of control the events of mitosis, and it has a way to shut itself off. Okay. So now within the M phase, you've got the spindle assembly checkpoint. So what you've got now is a way that the cell can make sure that the mitotic spindle is actually forming and attaching to these chromosomes so that we can pull these chromosomes apart, because really that's the idea of mitosis. We don't want cells to end up with an inordinate amount of chromosomes. We want them to have the exact amount that they need. So what this checkpoint does is it basically is going to sense some tension from the mitotic spindle, kind of pulling on those chromosomes that are held together along their lengths by those cohesin proteins. And eventually, as those cohesin proteins are breaking down and those cells start to pull those chromosomes apart, once they get pulled apart, now the events of G1 can start taking over as that cell re-enters into interphase there. Okay. So in the next presentation, what we're going to talk about is some of the effects of when this mitosis goes wrong, DNA replication goes wrong, and we get cancering and things like that. So if the cell cycle doesn't function properly, it leads to problems, and that's going to be the subject of the next discussion. Okay. So as always, if you have any questions, make sure you write them down. We'll talk about them in class, and we'll see you soon.